to set up the problem. Okay, so now that we've set up the problem, which is that people are doing these studies in convenient samples, but that these convenient samples are turning out to be quite like very much convenient samples that do not represent sort of the policy and practice communities that we care about. The rest of my talk is going to be sort of what I'm usually talking to people about how we could do this better. And some of the reason that I'm presenting this to you, I guess, as a statistics community, is a lot of my approach is really an approach that's somewhere between what is statistically optimal and what I think people will actually do if we're wanting to sort of to push practice to be better. So the first thing I'm going to talk about is how to plan better studies. Okay, so everything I'm going to talk about here is actually something you would get in a basic survey sampling class, right? So I'm going to say, well, first of all, let's actually ask these questions and think about what we mean by generalization. So we need a target population. We need to think about the eligibility criteria for the study. We need to think about estimands. What are we trying to estimate here? Any resource constraints and recruitment strategies, and sources of treatment effect variation. Now, this is, for those of you in the medical field, this is slightly different. I know in the medical field, people are actually usually not necessarily great about the target population part of this. There's a whole lot of problems there too about the lack of representativeness or similarity between samples and populations. But I do know that eligibility criteria tend to be much more explicit in medical trials than they have been historically in the social sciences. So I'm usually emphasizing that target populations, first of all, we need to have data on this top target population. So we need to be able to enumerate and make a list of all of the sites and have very clear inclusion or exclusion criteria. So I'm just giving an example here that we might say something like the study seeks to determine the average treatment effect in a population of 80,000 regular public elementary schools in the US. And so we're being clear that we're excluding alternative schools and federal schools, but we're only talking about schools that are public schools serving K through 6. And again, the idea is supposed to be that if we can do this very clearly, then policy and practice, people can understand if they're in this population or not. The next part you need to do is identify key moderators. And the reason for this is that random sampling is going to be very difficult in this case. So we're going to have to really monitor the kind of sample that we have. You might think of it as we are anticipating a very high degree of non-response. And so the moderators that we might care about, well, this is a circularity problem that's going to keep coming up in this talk, that in order to know, so that we need to know how the treatment effect varies in order to monitor and target recruitment in a way to make sure that we get a representative sample on those moderators, but actually we don't really have a good sense of why and how treatment effects vary. So we're like constantly in a circle here. So some of this is limited by the data that's available. This is definitely an area in which people are currently researching and developing methods for determining moderators in existing data and trying to understand how to best figure this out. What we often have are demographics. Those may not be the right variables. And I think the fields, the social science fields, at least, maybe I'm speaking from the area I know, definitely has some work to do, I think, in theorizing and understanding why they would expect effects to vary. So this is just an example from an old paper of mine where we're just using data from, this is the Common Core of data as well as data from the American Community Survey for an intervention study. So thinking, okay, well, like at very least we've got some racial demographics and free and reduced lunch, which has to do with student SES as well as various characteristics of the community. And then the idea is to divide the population into strata. Again, I feel whenever I teach intro stats or any stats class, really, I guess I'm always saying there's like a handful of tools that we use. Stratification is obviously one of those. It both decreases variation, residual variation, and increases similarity. Here the problem has been that these samples are going to be small. So we're not talking big survey samples of thousands of schools that are going to be chosen. We're talking samples of maybe 20 to 40, maybe 60 schools that are going to be selected. And so we need to have a small number of strata. And so here I proposed at one point that you could use k-means cluster analysis to do this, that you just take the variables that we've got and we look to see if there's sort of an underlying structure to these variables. And you can take many variables and reduce this down to a single dimension, and so have a small number of strata. And again, the idea of cluster analysis, right, is that it creates homogeneous clusters, which is the same idea as strata. This is from a web tool that I got funding several years ago to develop to help people do this, which is called the Generalizer, and it's just showing, a heat map of kind of an example case study of dividing it into 6 strata based upon these schools and sort of helping people characterize this. And then the idea is that you need to recruit. So you could use proportional allocation, and if 20% of the population's in one stratum, then you should recruit 20% of your sample. There's a big question of how to actually recruit within these. One strategy would be obviously to do this randomly. I have found it incredibly hard to convince people to do this randomly. Another strategy would be to use a more balanced sampling, a model-based sampling approach, which I proposed in a paper, which is to sort of recruit the most the stratum average schools, give priority to those. And then I think what people often do is some combination of maybe one of these, but also using convenience. So they might really have some set of schools they already know they're going to be in, and then they're looking for others. And then collecting information on recruitment. This is one of the biggest places that I'm always emphasizing to people, that we actually have very little information on what's happening in recruitment, and so it's very hard to understand what the sort of how much bias there may be. So this is just really getting people on board with actually collecting information, thinking about the strategies that they're using, thinking about collecting additional information during recruitment to understand why places would say no. And then this is just a kind of a short list of some of the range of studies that I've worked with, and that have, so I'm just kind of showing here that this is a range that like includes studies in various parts of our country, community colleges, high schools. I've got some in here that are using a combination of purposive sampling and probability sampling, some that are using probability samples. So it is possible to do this work, and it's becoming increasingly important, I think, in the field of education. I'll talk at the very end of this talk about a guide that we just wrote for the government that I hope will help. Okay, any questions before I move on? It's very weird. I always think to give a talk to just my screen and not have any sense of anybody else. Any questions?
None have shown up yet that need to be
Keep
passed
going.
on. Sorry. Yeah, no, it's
right.
good.
All right, I'll keep going. Feel free to interrupt.
Oh, yeah, Charlie.
Are you going to speak a little bit about some of the tools that you talked about developing?
Yes, I will. I'll come back to some of those.
Right, thanks.
Yeah, thank you. Okay, so what can we do about it? Well, one thing we can do is so let's say you already did your study, or you're even in the process of doing your study. There's a whole nother set of tools here about how to assess similarity and how to adjust for this. And so the assessment of similarity, so this is like, I did a study, and now I have a sample, and I have some potential moderators, and now my question is, well, how similar is my sample to this population, or to this population versus that population? And how do I characterize that to people in a way that is helpful? And so one strategy, of course, is you can calculate, you have these moderators, you can compare the sample to the population on each of the moderators and sort of say, well, they're similar in this way, but not in this way. When I started doing work in this area, I started thinking like a global measure of similarity would be quite useful. And so that's what this is. This is a global measure of similarity. The idea is that you calculate what are called propensity scores, which are, I always think, like a fancy way of saying probabilities. So we do this logistic regression where we have our sample data and our population data, and we're comparing them on a host of moderators using a logistic regression. So like a dummy variable indicator in the sample, and then you can get these distributions of these probabilities, of the inclusion probabilities, which you can put on the logit scale, which kind of pulls it out a little and normalizes it. And so I'm just showing you an example here that says, like, look, these two don't look exactly the same. We're seeing a long tail in the population. That's the solid line, which indicates that there are some parts of the population that are not being sampled. And so a question becomes, well, how could I characterize this distribution, or these two distributions, and how similar they are? that's what the idea of this generalizability index is that I proposed, which is just a global measure of similarity. It's really, you could think of it as a geometric distance between the two distributions. It has, I have a paper in which I note that there's some nice properties. This is actually based on something called the Badocaria distance measure, or Badocaria coefficient. It takes values between 0 and 1, which is really nice. It works even, it doesn't require them to have the same common support. And I always tell people you can sort of multiply it by 100 and interpret it like this, like something like the sample of schools in our study is 90% similar to the population of schools in terms of these moderators. So this, just to give you intuition, these are like histogram, just, you know, these are just variations of this to show you, like, look, when they're really separate, the index is small. When, as they become, you know, as the distributions become more similar, the index becomes closer. From this, I developed some rules of thumb based upon some simulation studies to say, you know, for the kinds of samples that we see, like sort of, you know, red light, green light here, like if it's really high, then like you might be okay using your average treatment effect estimator and not worrying, you don't need to do any adjustment. And then if it's really low, these simulations show that what happens is that there are huge parts of the population that are not represented in the sample, what would be called a coverage error. And that means that you can't really adjust. You can't adjust for something that doesn't exist. And so if you're in this middle ground, you can adjust, and there's a host of adjustment methods I'm going to very briefly go over, but they come with a pretty big penalty in terms of precision. So here, this is my preferred way of showing this to just sort of people, which is I can divide something into strata. I'm showing you the proportions in the strata versus in the population versus the sample, and I can reweight this. So I'm reweighting this. It means I'm going to upweight some parts and downweight other parts. And the more I'm upweighting or downweighting, I'm going to see that effect in particular on my standard error. There's a whole host of more complex adjustments out there. You could post-stratify based upon the propensity scores. You could also use inverse probability weights. There are some doubly robust approaches that you could use, which use a combination of weights and regression. Again, the biggest problem in most of these cases is the small sample size, because our recruitment process is on the clusters. So we have a small number of clusters, and so it becomes tricky. You know, one very large weight can really have a big penalty, and so there's a lot of bias variance trade-offs here. So just as a few caveats, the adjustment affects both the average treatment effect and its precision. They tend to be less precise. Obviously, the more similar your sample is to the population, the smaller the adjustment, which is incidentally what led me to thinking about, like, how do we get people to get better samples? Because getting better samples is certainly better for us than doing a whole study, finding out the sample doesn't represent the population, and then basically ending up with something that's underpowered. And then the last thing is that you have a lot of trouble with the fact that we can only adjust for moderators that we have in our data. And so I'm always encouraging people to be very careful in how they talk about these and the limitations of their analysis. I should say maybe right here that this is an interesting problem that I think probably occurs in other areas, which is if you don't do any adjustment at all, people often find that more trustworthy than once you've started doing these adjustments. So if I don't do any adjustment at all, I have to remind people I don't see the generalization problem. I only see the sample in front of me, and I can rely on the randomization within the study to focus only on internal validity. And the only way that I generalize this effect or transport it even is if the effect is constant, which is a huge assumption that the effect is constant that we've never really tested well in the study. But once I start adjusting, all of a sudden, as soon as I add one adjustment in, people begin to become worried that I have the wrong variables to adjust. And so therefore, I shouldn't trust this as much. So this is just like a little bit of like a puzzle that I'm always working with people to think about how sort of we have these hidden assumptions and how it makes us prefer simple things that actually have much bigger assumptions in them than the more complex. Okay, the third thing I'm going to talk about, unless there's questions, so feel free to jump in, is yes.
The second bullet on the final point, can you elaborate on what that means?
Yes, so when I have to reweight, when I reweight, what that means is the more different they are, you can think about there's going to be more variation in the weights. And when there's more variation in the weights, it ends up increasing the standard error of my average treatment effect estimate. So as the sample and the population become more different and I'm relying more on statistical adjustment, it means that my post-stratified sort of adjustments or my inverse probability weighting is going to end up inflating my variance. So I'm going to get something that's less biased, but that could potentially have very large.
Right. And now when I was pointing out, thinking about the potential moderators may not be actual moderators. Is that what you were addressing?
Yes. So this is actually, so the reweighting is on the potential moderators that we have data on. This is actually, I think,
Oh,
yeah. So we're using the moderators that we have to develop those weights. The concern becomes, right, that like if I include a moderator that is not actually related with the outcome, I could be not really changing the bias because it was unbiased or like low bias to start with. But now I've got paid this penalty of increasing the standard errors.
you're adding noise.
You're adding noise, exactly.
Okay.
And so this is an area where people I think are working right now and thinking about these trade-offs. Like I don't know what they are, so is it better for me to adjust or not adjust in this sort of low information situation?
Okay. Now I get the idea. Thanks.
Yeah, you're welcome. Okay, so this whole talk kind of goes in the order of my thinking too. The third thing that you could do is to actually think about this moderation and this heterogeneity. So as I've been doing this work, you start with, okay, the average treatment effect in my sample may be different than in the population. So then if I think treatment effects vary, then I should probably adjust I should either adjust for and get a better estimate of the average, or I should sample in a different way to really represent. But as soon as you start going down this path, I think it's hard not to start thinking, why am I so focused on the average? Like, shouldn't I care about the variation in effects? Like, wouldn't I want to know if treatment effects vary and be able to explain to people that you would expect bigger effects under these situations than under those situations? And so it leads you down this path of, okay, what would it look like? Maybe we should be planning these studies not just for the average, but to actually be testing hypotheses about variation. So I'm just setting us up here. I'm saying, look, okay, let's say I have, this is a study in which I've got maybe students in schools, and I can imagine potential outcomes. So if you have every student within schools, or every school has a potential outcome, which has, so I can talk about, we have an effect size that has to do with the average, but you can also imagine for each moderator. So if I think the relationship between X and Y is different in the treatment group and the comparison group, then, or another way of saying that is there's treatment by covariate interactions, then I can imagine that there's like a vector of these covariates, which I'm going to call delta here, which are these sort of the treatment interactions and how different these are. Something people like to do in randomized trials a lot when planning is to talk about the minimum effect size. And so we often, as you go back, we often talk about the minimum effect size for minimum detectable effect size for delta 0. So when we're planning, we say, well, if the sample size is this and these other design parameters are that, then what's the smallest possible true average effect size that we would be adequately powered for? You can do the same thing for what's called the minimum detectable effect size difference, which has to do with these moderators. And there's been some prior work in this area that shows what these are. I'm just showing it kind of in one case in which you've got students nested in sites. J here is the number of schools, N is the number of students within schools in a balanced design, P is the proportion in treatment. But when you break this down, there's a numerator part to this that people often explain away in previous papers. They've just sort of said, let's assume the sample and the population have the same variance in the covariates, and then this numerator disappears. And it occurred to me that that doesn't make a lot of sense. If I know the samples are different than the population on average, they're probably different from the population in terms of this variation as well. And so let's think about this as a design parameter, not as something that we just assume away. And so you can extract out, rewriting this, what I show is you can extract out this ratio, which is the ratio of the standard deviations of the covariates, these moderators, in the sample versus in the population. And so I call this R, like it's just a ratio. And so it becomes clear that, so for example, we might say, well, what if we have a homogeneous sample, a sample that does not have has less variation in these moderators in the sample than in the population, or a representative sample in which indeed they are the same, or a heterogeneous sample in which we have more variation in the sample than we do in the population. And so just to, again, root this back in some real data, I took some of the data that we had collected from this previous study of 34 schools, and I went back to look at several, these are, again, these might not be the right moderators, but they are the moderators I have data on. So this is total number of students in a school, student-teacher ratio, and then percentage Black, Hispanic, free and reduced lunch, which is like a socioeconomic status, and then urbanicity. And all I'm showing here, this green line across, is showing that would be if they were representative samples. And what you can see is that most studies actually are really homogeneous. They're quite homogeneous samples, which makes sense when we think about, if I'm focused on the average treatment effect, think about everything we know about lab studies, that in lab studies, you control the variation in order, get rid of as much variation as you can in order to get the most power for your sort of average treatment effect or your mean effect. Well, it turns out that that exact approach is great for the average, but it's really terrible, it turns out, for our ability to test hypotheses about moderators. So a simple way to improve power here, if I'm thinking in a design-oriented way, is I just need to recruit samples that are more heterogeneous than what is typical. And so at a very least, defining a population in advance and then trying to get a sample that's actually representative of that population, not just in the average, but in the variation, is important. But you also see, I could even purposefully, for the same fixed sample size, try to over recruit at the extremes to get a more heterogeneity in my sample. And this is the idea, really, of you think about of response surface modeling and optimal design methods that are much more common in engineering, in engineering sciences, where you're in a much more controlled environment, and you say, I have a limited number of design runs. I can only do a certain number, and so how do I do that in a way that is optimal?
Doesn't this increase the cost of the study, though? I mean, these things sound like they're the harder ones to get. So you have to look at how much you're going to pay for this data.
Yeah, quite possibly. And the question becomes I think there is a question of cost. Sometimes what I have found is you can actually, if we don't think it's really regionality that matters, you can actually get quite a bit of variation sometimes within driving distance of where you are. So if we think it's these moderators or others, you actually can, like, without traveling all over the country, that you can actually get fairly demographically like variation in your study without increasing your sample size. Now, anything I'm going to tell people to do, by the way, whether it's this or for the average treatment effect, is increasing the workload theoretically,
but giving you better results.
Yeah, you're getting better results. That said, I have a paper with a colleague who actually implemented one of these plans for the average treatment effect, and our paper is just sort of a case study. And he works at a research firm, and he was kind of recounting what happened in the study, and he kind of the conclusion was like, it didn't really involve more or less work than recruitment in general, because recruitment is hard, and people actually don't really, there's not really currently a state-of-the-art practice or a standard practice for recruitment. So very often people are just like throwing something at the wall and seeing what sticks, and so this strangely can provide a little bit more direction than is possible.
Thanks.
So de-optimal designs, really, we're just talking about reducing, what does that ratio become in a multivariate way? It has to do with this sort of inverse variance being minimized. And the nice thing is this idea of de-optimality has been around for a long time. So there's existing algorithms in place that you can use. So I'm just kind of cutting to the chase here to show like an example that I have in a paper that is showing at the bottom row. So these are each covariates. There's 5 variables that are potential moderators. Those are the columns. At the top, you have the population and what the population distribution looks like. In the bottom, you have what they actually had done in their study, which was quite a while ago, actually. And then in the middle, it's showing what a d-optimal design would look like. And so you can see that what it's doing, it's pushing you towards the extremes. It's pushing you to get more variation in your sample than you would get in the population. But because things are correlated, it doesn't push everything to the extremes. You still get quite a bit in the middle too. And so the nice thing is, because one of the things I often hear from people is if I said, I want to increase the heterogeneity of the study, they say, oh, now the sample is going to have to get to be a lot bigger. And so that's costly. To increase the sample size is bigger. And my point has been, you don't, even holding the sample size the same, you could actually dramatically increase your power. Okay, so the last part of this is sort of what can we do about that? What can we do about this? We can report generalizability clearly. And this is a place that there's a lot of room for growth, I think. So one of these takeaways is I'm always telling people when possible, try to have these hard conversations about generalization at the beginning of the study, not at the end when you find out it didn't go so well. And so, as a statistician, when I first got involved, I would say, oh, okay, so people will tell me what the target population is, and then I'll tell them like what they should do better or how to do it, get a better example. It turns out that actually asking people what the target population is not, they're not ready to answer it. Very often as a statistician consulting on projects, I spend a lot of time with people trying to figure out what the point of what they're doing is. They know maybe they have a contract, and so they know that they need to evaluate something. Or maybe it's a grant and they know they want to know if this intervention they're developing potentially works. But very often the people who know a lot about program evaluation they're not demographers, they're not survey samplers, they're not people that have a good sense of what the population looks like or what populations might matter. And so a lot of this is really helping people explore and think about, well, what would they ideally like? Who do they think will use this evidence? But also what is possible in the study that they have. So a lot of this also is helping people be resourceful and thinking about data that might be out there for developing population frames. Another sort of place to be clear is to be collecting information on recruitment. So if you notice, I did all of these analyses where I showed samples versus populations. The difficulty in those cases is you can imagine that there's actually a 2-step process here. So there's 2 selection processes. First, researchers had to actually have to ask places if they would be involved. So that's like selection number 1. And then conditional on that, if they were asked, they could say yes or no. But what I see when I just look backwards is only those 2 are collapsed. So I don't know when schools are not represented in studies, if they're not represented, if sort of parts of the population are not represented because researchers didn't ever ask them to be in the study or because they didn't want to be in the study. And you can imagine those actually have 2 different kinds of effects. Like if researchers don't ask, that's one thing. But if you're asked and you say no, there might be information in there like the intervention is not palatable to you, it's not of interest, it's not, maybe you already have something like that in your school. There's a variety of reasons, but when we're getting to sort of bias and the average treatment effect or what this means for interpreting the findings, we kind of need to be able to separate those 2 out. And so a lot of this is sort of encouraging people to actually collect this information and then to try to provide some basic analyses showing what these different processes look like, the kinds of places once contacted, if they say yes or no, sort of what's going on there. The third one is to sort of always suggest to people at the end that they should, I guess there's 4, but one of them is being covered up. Compare your sample to relevant target populations and adjust for differences. So what I have here on the left is another figure from the Generalizer, and I'm going to say a little bit more about that in a second. And the Generalizer web tool, you can actually put in the school IDs, and it will compare the sample that you have to a population. and it will produce this nice little map that says, it compares it for each state to say, okay, your sample is similar to this state, but not to that state on these moderators, to give a sense of where results may apply, where your average treatment effects may apply confidently and where it might not. And so the idea is that you should be reporting to people, which is number 4, help people understand where they apply and where they don't apply. So this is being very clear, like this study, these findings, I feel confident that they represent this population, but this study doesn't touch on that population. This is actually a hard sell because there's sort of, I think, some sort of psychological process in which people prefer general statements over specific statements. But if you think about the replication crisis that is occurring in the social sciences, a lot of that replication crisis is because people make these bold claims that things are going to work everywhere, and then they test them in a very narrow spot, homogeneous sample, and then later studies in other places figure out that it's not the same thing. And so it becomes, you know, it was like really from the beginning the claim was too bold. It wasn't ever ready to be able to share it in that way, be shared in that way. And then, okay, so then the last thing, and then I really am going to hope that you guys have some questions, is resources to help. And so these are sort of 3 things that I've been at work on. So one, I have a picture over here to the side of a recent guide, which I have a link up here for, and I can probably drop it in the chat or something if it's easier for you guys, which is, so the Institute of Education Sciences commissioned this white paper, a toolkit on generalizability, and my co-author Rob Olson and I wrote this. It's like 40 pages ish, and it includes I think 7 recommendations, kind of more detail on a lot of what I've said, and that the idea is that this is available to anybody planning studies, planning or analyzing or conducting evaluations. My hope is that by them putting their seal of approval on this, it will encourage more people in the evaluation community to actually start paying attention to this. The second thing is a web tool called the Generalizer. I think I got funding for this in 2015 or 16 from the Spencer Foundation, and then another grant from the Institute of Education Sciences to continue it. And so my original idea here was that that people weren't going to actually implement any of this stuff if they were writing grant proposals. Like, when do you need to have a plan for recruitment? When do you need to make a sampling plan? When you have no funding. Because if you wait until you're funded in order to do this, it's too late. So you didn't budget for it in time or money. So the idea is you have to plan it at the beginning, but you're writing a grant proposal, you don't know much about it. And so the idea was to create a tool that would, I like to say, in less than an hour, you can go through, define a target, if you're in education, K through 12 or in higher education, you can define a target population and then select moderators from existing data and divide your population into strata, and then it brings you all the way through the end where it gives you a report language on how to talk about this, as well as CSV files that if you download, that you get over email. When you get them, you open it up and it's got a list of all the schools in each stratum and their contact information, and by default some columns for you to keep track of the information during recruitment. The web tool also has a part where you can do assessment, where I was showing you the maps. So it's going to show you how to, you put in the school IDs, and then you go through this process, and it will help you create this map. And then there's a new part of it that's more recent, which is power analysis. And so this is, right now, it's just power analysis for the average treatment effect. But it covers a wide variety of designs, cluster randomized, random block, or multi-site with various different levels. And so it's supposed to be now like that you go there for like all of your design considerations when planning. And then I'd say another feature of this is it's web-based, and we've really invested a lot in the user experience of this. I think this is very uncommon in statistical software. It teaches, so it's not just intended, you don't need to read a booklet, you don't need to read documentation. The idea is you can sit down and it will teach you the things that you need to know in any one of these things. It will sort of explain them to you and guide you through it. And then the last thing is there's a new R package. It's not quite ready yet, so it's still up on our GitHub, but I think it'll be ready soon, that implements all of these methods for planning, recruitment, assessment, and adjustment, all in an R package for those that have other data or in other fields, like, you know, that are not education, or that want to have more flexibility. And that is it. Thank you.
Thank you. That's great stuff, Beth. I don't have any immediate questions in the Q&A, but I've got some. I'm wondering if this plays into political polling at all in terms of generalization and why people can't get their estimates right.
I mean, I would bet so. I would bet so. I mean, the problem, so when I first started doing this stuff, I felt a little bad about proposing things that were not just simply random sampling, because, you know, it's sort of everything we know from my, you know, from my training in survey sampling, like we should be doing things randomly. But non-response has become a big problem in polling, right? It's a problem in polling. It's a problem in sampling in general. Non-response rates are up. And you're right, political polling, where people are choosing to poll, is different, you know, obviously can lead to different results. So I would expect similar problems there.
And do you know if anything like this was done in terms of the COVID vaccine trials? Clearly, they wanted to know about heterogeneity of effect there. And
That's a great question. I don't know much about how they selected people in to those trials. I remember thinking about this a little. I don't know if you remember early on when they were, I guess, midway through, somewhere in that like long several years. They were comparing the effectiveness or efficacy of different vaccines, and it became clear that it's actually very hard to compare the efficacy because they were tested during different periods of the pandemic. So you had Pfizer that was tested early on, I believe, and Moderna was tested later, and it became clear that if you were tested early versus later had to do with what was out, like what variants were out at the time. And so I remember thinking like, oh, this is exactly the same problem, that like the efficacy of this vaccine depends upon the variant and the time period, because I think it also was like one of them was tested more in the winter and one of them more in the spring.
that's great. That's great. I think of I thought of in the middle of this talk, I was remembering a tweet. This is probably a bad place to go off on, from Frank Harrell, about somebody had asked, you know, how do you report the effect in the treatment arm and the control arm? Do you use the mean and the standard error, or the mean and the standard deviation, or whatever? And he wrote back, neither. You just report the difference because it's not a generalizable study. And I'm thinking he probably should have made sure that the sample wasn't collected as you've been advocating. Maybe it is a generalizable study. But anyway,
It's interesting. I have found that some statisticians really want to hold close to the internal validity, and they say, I don't like, the nice thing about an experiment is the randomization makes this unbiased, and I don't need a fancy model, and I can rely only on the randomization. And so the idea of modeling or thinking about how the sample relates to a population, unless you have a random sample with no non-response, I don't want to do it. But then I'm always left with, but okay, so, but when do we talk about the results of these as only applying to the trial? Like if we, by design, are doing these studies in the field so that people will make decisions and they could end up in clearinghouses, aren't we admitting that we think it matters, that we are trying to generalize? And it becomes, to me, like a matter of, is your model better or worse?
And aren't you really emphasizing that in your statements about the selection of the responders, that it's not even really a randomized sample? It's just what you've selected. And
yeah.
And the other point that I think I mischaracterized earlier by saying that it's costing more. I think what you're doing is, if somebody wants to sample, it's a more effective way of spending the money.
Yeah, exactly.
And then maybe instead of adjusting, maybe they're more open to sampling a little bit more in a spot, and then they don't. I mean, it's sort of like they have an option of going either way.
Yeah. Yeah, I think that's exactly right. That's one of the things I'm always emphasizing to people It really is interesting to me though. Sometimes if I say, well, you might oversample this group and oversample that group to get these more extremes, people will start be like, well, but then I'm like choosing the sample in some way. Whereas the current practice is like, if you just don't think about it, you don't have to worry about this problem. You can be much more comfortable with it if you never really think about how this.
That's a head in the sand.
Yeah, it's a really a head in the sand kind of problem. I was going to say the medical example I have. I was at an NIH meeting several years ago, 5 or 6 years ago, and I heard somebody say that I mean, this is definitely a problem there as well. So the average age of a person with pneumonia is like 65. The average age of a person in a trial for a drug treating pneumonia was 30.
Wow.
Because in medical trials, so in education and the social sciences, these eligibility criteria, like we really don't understand clearly why some schools are in studies and why some aren't. Nobody actually is very clear about that, what their selection mechanism was. But in medical trials, there's usually explicit, like very explicit inclusion-exclusion criteria, and they want to exclude comorbidities. So, which is tricky. So another case that somebody said was if they're trying to test a drug for an antidepressant to treat depression, that that often inclusion-exclusion criteria will exclude people that have anxiety because they only want people with depression in the trial. But the problem is that anxiety and depression are highly comorbid. And so most people who are going to use the antidepressant are actually probably also dealing with anxiety. And so what's ideal for us to understand understanding for a mechanism and as a scientist is at odds with sort of the use for people.
Your point about choosing 30-year-olds to test the efficacy of a pneumonia drug, I mean, learning that a drug helps 30-year-olds get better, it doesn't give the 60-year-old very much confidence as well.
Not at all. But we do. This is what I mean. Historically, medical stuff does this, right?
Wow.
That we do the trials, and so the meeting I was at years ago was about extending the life's course. So getting geriatric patients and neonatal, so children, all the way up, like children and geriatric patients, to be more commonly found in medical trials, because those are populations that use medicine, but are very often have been historically excluded from a lot of medical trials.
Well, it sounds like people are going to think about turning some rocks over and seeing what's underneath, here with this, what's in. That's really good.
I mean, I hope so. I hope it's a place, personally, I think it's a place in which, it really is this, like, what are we doing with the evidence? Really, like, it's different. It's very clear to me that if you design systems for understanding, that will not necessarily get you to use, but we often sort of think that understanding immediately applies to use. And I think it actually leads to distrust of the scientific community.
Yeah.
When people realize, like, oh, like, I'm not represented in this trial. I'm not represented in these studies. These studies are only on healthy volunteers that are men, or, or they're only in this kind of school. It leads to people sort of not trusting that, that the science is important to them too.
I'm glad you emphasized. That's a really good point. Thanks.
Well, Beth, thank you very much for joining us tonight. I learned a lot, and I'm curious to think about how it's going to apply to the medical stuff I work on. I've got some big conversations to have with some of my investigators.
Thank you. Thanks a lot. It was nice to talk with you.
Yeah, okay. Thanks again, and thanks for joining us, everybody. I'll put the